starters, for those of you who are paying attention, this is drastically different than the talk I was planning to give when I submitted my abstract. So apologies for that, but the impetus behind changing this is that we recently got the most cutting-edge DNA sequencing machine on the planet. I've been sort of living underneath that thing since we got it, and I was just too excited about these results to talk about my old stuff. So today I'll be talking about long-read sequencing. And I'll start this talk by sort of giving the sort of industry standard for how whole genome comparative analyses are done, talking about one of the sort of challenges that you face if you're using this type of technology, and then obviously propose how long-read sequencing can help ameliorate this issue. So for starters, let's just use a sort of cartoon example. Let's say you want to do some comparative genomics for your favorite organism. One of the first things you would want to do would be to generate what's called a reference genome assembly. And a reference genome assembly is essentially just like a roadmap that tells you where the given sequences in the genome are and where they are on the relative chromosomes. And although this is becoming easier to do, this is actually a relatively intensive process. It's expensive and bioinformatically challenging. But the reason it's worth investing the time in doing this is that once you've done this for a representative individual from your species, then you want to do some comparative analyses. Let's say there's a population that exhibits a blue phenotype, and you wanted to identify the blueness gene, wherever that might be. Rather than going through the whole genome assembly process again, what you could just simply do is a process called whole genome resequencing. And this is typically done with a type of sequencing called short-read sequencing. And short-read sequencing is exactly what it sounds like. They're base pair sequencing that range from around 150 to 200 base pairs in length. There's a little bit of variability there, but this is like the standard. And what's nice about this is, since you've already resolved the genomic architecture that is relative where genes are relative to one another on each chromosome with the reference genome, you can just align these reads to the reference genome, identify single base pair mutations, also known as single nucleotide polymorphisms, and then you can use these genome-wide SNPs to do some really powerful analyses, such as phylogenetic analyses, or if you were to want to identify this blueness gene, you would do what's called a genome-wide association study. And this is just a method for identifying specific single base pair mutations or a group of base pair mutations that are associated with certain traits, in this case, blueness. However, it would be really difficult to overstate just how impactful this type of analysis has been. Basically any field with the word genetics in it, this has been a very powerful tool that's led to a great deal of insight. There is one major challenge to this, and that is that depending on what you're sequencing to a reference, where you're at to a reference, anywhere from 5 to 40% of these reads are either improperly mapped, meaning they're being lined up to a part of the genome where they don't actually belong, or they're just not mapped at all. And this typically occurs if there's a part of the genome that's missing. And so, structure changed a little. And so what we've learned from if we were to look at the genome of an individual from this different population, what we find is that these improperly mapped reads actually belong to a region of the genome that is actually biologically missing from the individual that was used to make the reference genome. And this is a type of this falls into a certain class of genetic mutation known as genomic structural variations. And we can define these as mutations that affect over 50 base pairs in length. And structural variations come in sort of 5 different flavors. I only have 4 of them listed here, partially because I couldn't come up with a clever idea of how to use the cartoon to display one of them. But if we look at the top, this is just sort of the ancestral chromosomal state, and the reason it's relevant to show that is because if you're going to claim something as a mutation, it has to be a derived state essentially different from the ancestor. And that top row there are these single nucleotide polymorphisms. These are things that we can identify really confidently with short reads. But these class of structural variations, deletion mutations, duplications, insertions, and inversions, hopefully those are sort of intuitive based on the cartoon, these are larger mutations. And so these, just by definition, are actually harder to identify with short read sequences because it's harder for a short read to map across the reference, across the variation itself. And I'll explain that in a little more detail in a minute. And the reason these are important types of mutations to identify is, for example, if we were to compare any 2 human genomes in here, we would find around 3 million to 5 million single base pair mutations. However, we would only find about 2,000 to 2,500 structural variations. But the difference is that in those structural variations, around 50 million base pairs are likely to be affected by structural variation. So these types of mutations are actually affecting a much larger portion of the genome than single base pair mutations are, and as a result are more likely to actually interfere with how certain genes function. So they're really important genetic variations that are really hard to detect using just short And so if you can guess from the title, the proposed solution is going to be long-read sequencing. And in order to sort of exemplify how it is that long reads can help ameliorate this problem, I'm going to use an analogy. An analogy, which you may have heard a million times, is sort of like a puzzle. So here, if we take this beautiful piece of art from Tom Larry, and pretend that it's the cover of a puzzle box, and then the resequencing individual, the individual that we want to map to that reference. So if we pretend that the box cover is the reference genome, and the puzzle is the individual that we're using resequencing using short-read sequencing. We can see, first of all, the sort of elephant in the room, the blue chunks. I actually left these in here for an important reason. These are sort of representative of what we call repetitive DNA sequences, repeated sequencing motifs that are the bane of every geneticist's existence. We'll ignore those for a minute. Even if we just look at these easier to identify pieces, for example, these 10 segments that we see here, these small pieces that we can see sort of clearly have a chunk of the letter line on them, and clearly have some Charr spots above and below them. But they're similar enough that it's actually quite difficult to identify which of these individual pieces align to this particular part of the fish, part of the puzzle. Alternatively, if we were to segment this individual into just fewer but much larger pieces, we can then see how it would be much easier to reassemble what this lateral line looks like, or if we were to map these to the puzzle, which is analogous to mapping DNA sequencing reads to a reference genome. Each of these have much longer, they're much larger in size and therefore have their own idiosyncrasies that make them much easier to actually map back to the reference. And this is one of the methods by which, this is the primary mechanism by which long reads solve this problem. And sort of just close the loop on this analogy, we'll go back to our cartoon in this area where we have this genomic structural variation, and that we're having trouble to identify with short-read sequencing. And if we were to use long-read sequencing and map those reads back to the reference, one of the reasons we would be able to resolve this structural variation or identify the structural variation much more readily is because, as you can see, not only are the pieces larger, so we're able to identify where they go, but we'll have several instances where a single read, so a single DNA molecule that we're able to sequence, actually spans that inversion breakpoint. So in this instance, the blue bit would map really nicely to the reference genome, and then the orange bit, which is representative of the genomic structural variation, would have really low mapping quality or word map at all. And with that dichotomy within a single read is a very strong way of identifying. We can use that information, that dichotomy within a single molecule, to identify where these structural variations exist. And then you multiply that by however many times genome sequencing coverage you have. Okay, so enough flapping my jaws, let's get into some results. I'm going to give some examples of some of the works that we've been doing with lake Charr. We've heard a couple talk about lake Charr today, including a nice one by Dr. Craig just now. And so this is, there are 10 populations shown here on the left on the map. Yeah, on the left on the map, you can ignore the shapes for now. This is a map from a different talk. And these are fresh results, so I haven't had much time to make a new map. But these are the populations that we're sampling. And so far what we've done is for 9 of these individuals, we've generated long-read genome sequencing of somewhere around 10 to 15 times genome coverage. You can think of genome coverage as the number of lines that you saw sort of overlapping. And these reads have ranged from anywhere from 5 to 25,000 base pairs in length. And my average read length, so that's the read length M50, which is sort of a proxy for median read length. My average, for example, is around 10,000 base pairs in length. So if you remember, short-read sequencing was only around 150 base pairs, so these are much larger puzzle pieces that we're using. And in addition to that, we also have sequenced 3 individuals to much greater depth, so around 50 to 60, around 60x genome coverage. And we've used those to assemble de novo genome assemblies, which I'll explain a little bit more in a second. All right, so this is just sort of a bird's eye view of what we've seen. So here on the top, what we've seen so far, rather, on the top you have a list of numbers. Each number corresponds to a column, and each of these represents one of the 1 through 10 chromosomes for lake Charr. There's actually 42 chromosomes in the genome, but it didn't fit nicely on the slide. I'm just showing the first 10. And I should mention that we mapped these long reads to a really nice chromosome level genome assembly from Smith et al. That they published a couple years ago. And so the sort of general patterns we're seeing is that lake Charr harbor a lot of genetic structural variation so far, similar to around what we see in humans. I think the lower end of the more conservative side is around 1,500 genomic structural variations, affecting anywhere from 20 to 60 million base pairs in the genome. So this is likely to have a big impact on how these animals are sort of different in traits. So there's a lot here, so let's just focus on a couple of examples. For this, I'll just talk about the Cisco-wet lean lake Charr, Cisco-wet versus the lean lake Charr work. And for those of you who don't know, lake Charr exhibit pretty extreme phenotypic variability. And in Lake Superior, for example, there are 4 different ecomorphotypes, and they tend to diverge across depth. In this case, this is the deep water north, the Cisco-wet, and they've been shown through a lot of nice studies that they prefer deeper habitat, have larger eyes, and relevant to this point, they have much higher adipose lipid content. And so I sort of asked the question, of these genetic variations, I'm just going to focus on deletion mutations, because these are actually the types of mutations that we can identify with the greatest level of confidence. And I didn't have enough time to analyze all the other ones, so I started with this one. So I said, of these deletion mutations, or how many deletion mutations do we see in Cisco-wet that we don't see in Seneca reference? And of those, how many interact with genes? How many of these deletions took out a chunk of actual functional gene in the genome? And I looked for patterns in which genes were actually affected. And so here, what I'm showing is a metabolic pathway. It's sort of a cartoon version of how this thing called the hippo signaling pathway works. Each box here represents an enzyme, and the green boxes are enzymes that the gene of that enzyme, that codes for that enzyme, has been affected by a putative deletion mutation in Cisco-wet. So we identified about 200 genes that were affected by deletion mutations, and 7 of those occur just in this one metabolic pathway. And what's interesting about this metabolic pathway is it's been shown really robustly in both human and mouse models that disruption of this pathway leads to severe obesity. And so we think this is pretty strong evidence of how some of these structural variations can be leading to different phenotypic variation that we see among these different ecomorphotypes. This is getting to the idea of whether or not, it's not fully resolved yet, whether or not these different morphotypes are the result of pure genetic differences, or if it's environmental difference, an ecomorphotype, or genetic morphotype. So we just thought this was a nice example. Again, this is preliminary. We still have to do some more proofing, but so far I've been very conservative with what I call a confident structural variation. All right, my last example, I'll show how long-read genome sequencing can be used to assemble nice reference new genome assemblies. And in this case, on the y-axis, what you're looking at is a nice genome assembly that we've created for a population from Little Moose Lake in the Adirondacks, which I guess is now west of here, to the east of here when I talk about this. But each row in this plot here is corresponding to one of the chromosomes that we've assembled in the Little Moose Lake individual, and then each row or each column on the y-axis is the same chromosome in the Seneca Lake reference. And you see, by and large, you see this one-to-one relationship. And what this is reflecting is we're essentially mapping the DNA sequence of each chromosome from each individual to each other. And so you'd expect a one-to-one relationship. These deviations from the one-to-one relationship are actually reflective of several ancestral whole genome duplication events. And so this whole problem is actually extra hard in lake Charr because, like all salmonids, they experience a whole genome duplication event. And each of the chromosomes has another chromosome that a big chunk of the sequence is similar to it, so it's extra hard to map reads to it. But for the rest of the talk, we can just focus on this one-to-one line. So if we zoom in here just on chromosomes 34 and 35, if we look at chromosome 34, we see this is what most of the chromosomes look like. This one-to-one relationship. In here on the x-axis is the Seneca Lake reference genome, a chromosome 34, and then on the y-axis is the Adirondack Lake individual. However, if we look at chromosome 35, we see this deviation from the one-to-one line. And what this actually represents is what's called a chromosomal inversion. This is actually a 10 million base pair segment of chromosome 35 that's flipped. And this is really relevant because chromosomal inversions, there's a lot of literature coming out now that supports that chromosomal inversions actually inhibit genetic recombination. So they reduce the ability for genes to be mixed up in these regions. If two individuals that had different karyotypes or different individuals, one individual had an inversion and one didn't, and these types of chromosomal inversions are now being shown to really rapidly accelerate things like speciation events or rapid adaptation to local environments. And we've got some nice evidence that this population from Little Moose Lake is actually quite divergent from all the other individuals, which we sort of expected based on some work that Dr. Marsden and Dr. Kruger did with some allozyme work back in the day. And so this is just a nice example of how you would miss that you wouldn't miss, but it would be quite difficult to identify this type of mutation if you were just using short-read sequencing. And so, in conclusion, I just sort of a summary. Long-read sequences are less susceptible to what's called reference bias. And reference bias is simply when your comparisons of when two comparisons are being done between a genome are limited or biased based on whichever individual was used to make the reference genome. So, for example, those reads that were mapped poorly would have been excluded from many analyses. And then also, long reads allow for the incorporation of structural variations, which I hopefully I've convinced you are important biologically into conservation type work. And then they also greatly improve our ability to assemble nice chromosome level genomes. With that, I've got my audience to acknowledge, my co-authors, and the people that have helped me collect fish. And just as a teaser, we've generated the first draft reference pangenome. I'd love to talk to everybody about what that actually is.